Okay, let's get started. A couple of announcements before we get going. First one is this Thursday, we have another candidate for a position in the biology department, a new professor. Forgotten the name and the title of the seminar, but I can pulled it up and I'll keep reminding you again. It's on Thursday. Second thing is this Friday is not the midterm, but it's my midterm. Because the dean will be here, and also I think 4 students from the biology teaching course, so they're going to tell me how horribly I'm doing. As the case may be, we shall see. So they'll have some feedback for that, so there may be a few extra people in class, by about 5 extra people in class, so we've got plenty of seats for them to be here. Today we're going to talk about messenger RNA processing. Highly unlikely that we'll get through all of it. Those of you who looked at the notes will probably think, wow, he's either going to talk really, really fast, or he posted notes for basically a couple of lectures. So hopefully it's the latter rather than the former. But that does mean that a week from Friday is what? Is midterm 2 already. How did that happen? Quarters are just crazy that way, aren't they? Okay, so good practice for that is what? Exactly. So start out. Which of the following eukaryotic transcription factors do not have a homologue in archaea? Nucleosomes, TBP, TF2B, TF2H, or RNA polymerase II? Ten, 5, so which of the following eukaryotic transcription factors do not have a homologue in archaea? Nucleosomes. Is that a factor? Maybe, but he put it in quotes, so maybe he actually did mean nucleosomes. Some archaea do have nucleosomes, so they still have some of those issues. TBP? Is there TBP in archaea? Yes. What's the homologue of TF2B in archaea? TFB, exactly. What's the homologue of RNA polymerase II? RNA polymerase. What does that leave us with? TF2H. What does TF2H do? Helicase and it's a kinase activity which phosphorylates the C-terminal domain of the largest subunit of RNA polymerase II, which is going to be really important for the rest of today's lecture as well. Does the archaeal DNA-dependent RNA polymerase have a CTD? No, it doesn't. So both of those two activities would tell you that that's what's going on there. So today, as I mentioned, we'll talk about sorry, yeah.
It phosphorylates the C-terminal domain of what?
Oh, yes, so sorry. What does TF2H do? So TF2H phosphorylates the C-terminal domain of the largest subunit of RNA polymerase II. And we'll talk much more about that in the next couple of slides. So, and it's basically all about messenger RNA processing. So the M here is in parentheses because of this ribosomal RNA down here at the bottom. But messenger RNA processing in eukaryotes is basically 3 or maybe 4 main steps. The first one is splicing, which is taking out of introns but just as importantly, hooking exons together. So your classic gene, or here they have transcription unit in a eukaryotic system, has a few exons. If anything, there are more and bigger exons here than in a normal case. We'll look at some normal cases a little bit later on. So the vast majority of the RNA, which is being transcribed by the RNA polymerase II, gets chopped off. You also have a 5' cap that gets put on and 3' polyadenylation. Once all of this has happened, this messenger RNA gets exported from the nucleus and then undergoes translation, which is what we'll start talking about on Friday. Yes?
So splicing occurs with RNA polymerase, or is it a separate protein?
So the question is, does splicing happen with the RNA polymerase, or is it a separate protein? So what we'll talk a lot about today is that it's all happening at the same time. While transcription is taking place, all these modifications are taking place on the RNA, but it's different proteins that are doing that. It's not the RNA polymerase itself that is doing this. Just for comparison, down here at the bottom, I should have crossed out prokaryotes and just put bacteria, because we actually understand very little about what's going on in archaeal transcription and messenger RNA processing. Here, for the most part, your messenger RNA is made directly into protein, and it's a direct transcription of that particular piece of DNA. As always in biology, there are exceptions to this. And last time we talked briefly about what happens in terms of bacterial transcription and translation. As soon as the messenger RNA comes out of the RNA polymerase, ribosomes jump onto it and start translating it. So they're very closely coupled. Whereas in eukaryotes, transcription takes place in the nucleus, translation takes place out in the cytoplasm. So getting back to your question and a couple of these questions about the CTD, how is this happening? And the answer is, well, not how is it happening, we'll talk about it a little bit, but sort of when is it happening? And the answer is it's happening all at the same time. And this is for messenger RNA processing in eukaryotic systems. So C-terminal domain of RNA polymerase II, this is the largest subunit. Drawn here is a little tail, and the little marks here mean that this is a really long tail. And actually the structure of the CTD, what little we know about it, because it's really flexible and it's hard to determine the structure, what we know about it is it's probably at least as big as the rest of the RNA polymerase itself, if not bigger. And what it is a whole set of repeating amino acid sequences, particularly with these serines at the 2 and 5 position of this 7 amino acid repeat. Serines, of course, are great because they can get phosphorylated by what? TF2H, that thing that's not in archaeal transcription. So TF2H is phosphorylating that. It is important for promoter escape. And last time I mentioned that it was interacting with the DNA because you get your negative charges. The negative charges are repelling the DNA. It's probably actually more important for dissociating with the other proteins that are present at the promoter. You remember all those extra things, the mediator, all of the other general transcription factors, et cetera. So phosphorylation of the CTD is probably at least as important as, for undoing those interactions with the other proteins as it is in terms of repelling the polymerase from the DNA in that particular case. But from what we're most concerned about today, has to do with all of the other things that phosphorylation does for the C-terminal domain of the RNA polymerase, and that is an interaction with all of these other proteins, particularly capping factors. Again, what does factor mean? Protein. So these are all the proteins that are involved in capping. You could also call these the splicing factors here as well. Why they call one of them factors and the other one proteins, who the heck knows. And then also 3' end processing proteins, which of course are those going to be involved in the poly-A tail formation. So all of these guys together. So the RNA polymerase is making the RNA, but it's got all of these extra proteins, factors, again, whatever you want to call them, and the CTD, which is conveniently very close to the RNA. So as the RNA is being made, all of these proteins that are going to be acting on that RNA are right there, and not surprisingly, they modify it as this happens. So what do these modifications look like? Down here at the bottom, our eukaryotic messenger RNA, at least I remember to change this one, where we have a cap structure, which is a methylated G that gets added to the triphosphates at the end of the messenger RNA, the 5' end. Your coding sequence, which has usually been spliced, and then your poly A tail, which is added here at the end. In bacteria, you don't have any kind of modifications that happen at the 5' end. So this is just a triphosphate, exactly what the RNA polymerase used to start with. And then a messenger RNA. In many cases in bacteria, but by no means all, this messenger RNA can encode multiple different proteins, but they're multiple different proteins with these separate coding sequences. And it turns out this is really useful for regulation, and we'll talk a lot more about regulation after the next midterm. But we're going to concentrate for the next couple of lectures on the eukaryotic system, where it's almost always one protein that's being made from one RNA or a whole different family of proteins depending on the splicing processes. Terminology-wise, polycistronic, monocistronic, sometimes you'll see this. I'll probably not use it terribly much. But cistron basically just means one protein coding sequence. So here, polycistronic, multiple coding sequences in one RNA. Here, monocistronic, one coding sequence in one particular RNA. So let's focus in on this cap structure. What does it look like? It's something that gets made after about 25 nucleotides get transcribed. So after you've gotten past that abortive initiation step that we talked about before, now the RNA has come out of the big RNA polymerase complex. Then what happens is that RNA gets modified by enzymes which are hanging out on this C-terminal domain. And I just noticed that we've got moving this stuff around is confusing. Let me just get rid of this for now. Close that. Maybe this will help. No, it's still not helping. Okay. Be that as it may. So the arrow moving, it's a problem with these clicker questions. They overlapped everything, and I try to take up too much space. Okay, so underneath our clickers, it says phosphatase. See, phosphatase is losing a phosphate here. So as these 25 nucleotides get made, you've got a triphosphate at the end of your new RNA. That last phosphate, the 5' phosphate, gets cut off, and then you have a enzyme with my favorite name ever in molecular biology, the guanylyl transferase, which takes a GTP and adds it to the end of your messenger RNA. And this one phosphate right here, sort of shown here in red, comes from that GTP and hooks up to the two phosphates that were left here after this phosphatase activity. So what you end up with is a 5' to 5' bond, which is really kind of funky. We haven't talked about 5' phosphate bonds. But it's basically flipped over that nucleotide. Normally it's 5' 3', but here it's 5' 5' as they're hooked up to each other. And what that does is it gives you a very unique structure at the end of this messenger RNA, which is really important for, surprise, surprise, interacting with proteins. And this particular protein is called the cap binding complex. So we have our guanylyl transferase, puts this guanosine on here, but all of you have been watching properly. It's not just a normal guanosine, it's a 7-methyl guanosine, which then has a methyl group, which has been added to this position on the guanine. In many, but not all, of these transcripts which are made by RNA polymerase II, you also have methyl groups that are put onto riboses in the first couple of nucleotides in this messenger RNA that's being made. That's what gives you cap0, cap1, or cap2. Cap2 is 2 methyl groups on each of these riboses. Cap1 is 1 methyl group, and I give you one guess what cap0 is. No methyl. Exactly, perfect. 5 methyl groups. No methyl groups on any of the riboses. For the most part, cap2, and this methylation is probably helping the cap binding complex to associate with this end of your messenger RNA. And this is probably beating the dead horse here, but ends of nucleic acid chains are incredibly sensitive to exonucleases. Exonucleases are everywhere. They love to chew in at the ends of things. That's why we have telomeres in your DNA. That's why we have all the proteins that are involved with double-strand break, Rec-A, that binds to single-stranded DNA. All of these protecting of ends is a really important aspect of things. So the 7-methylguanosine and the cap binding complex are really the best things as far as making that structure which allows the protein to bind at the end and protect that 5' end of your messenger RNA as it's being made. Clear? Not clear? Questions? Yes?
So the methylation of the end of the primary transcript is targeting it for nucleosome activity? You said the methylation is attracted to endonucleases?
No. So sorry if I misexplained that there. So the methylation of the 7-methylguanosine, that's then helping the cap binding complex to associate. The methylation of the riboses is also helping that cap binding complex, but also is protecting against exonuclease activity chewing in at the ends. Yeah?
So after the methyl group is added to the base, there's another methyl group that's added to the ribose chain? Is that what you're saying after that?
Yes, exactly. So there are 2 still methylation reactions, so it's methylation that happens. But the methylation after the 7-methylguanosine, there's always a 7-methylguanosine. Every cap has a 7-methylguanosine on it. But some caps also will have phosphorylation on the riboses and at the 2' position. Probably the reason for that is 2' OHs, as we'll see as we keep moving on through the rest of the modification today, are really quite reactive. And so blocking that reactivity may also help. Other questions? No. And whenever he says that, what does he mean? We're going to have a clicker question. Of course. So how many different enzymatic activities, which is covered by my multiple choice there, are required for the formation of a cap on eukaryotic messenger RNA? Do not include the RNA polymerase or its modifications. Because I'm sure someone's going to say it's the RNA polymerase and TF2H. No, don't include those. Different enzymatic activities. Activities. 10. So what are those 3 enzymatic activities? 3 is correct, by the way. Phosphatase, guanylyl transferase, and methyltransferase or methylase. So why did I not put 4 on here? Because you could argue with me, which would be a very good argument, that the base methylation enzyme is actually different than the ribose methylation enzyme. So, yeah.
I'm just wondering, because if you're talking about the methylation of the nucleotide sequence as being one of the enzymatic activities, one of the options would
So the question here basically is if you would have a CAP0, for instance, which has no methylation of the riboses, you're still going to have methylation of the 7-methylguanosine, so there's still going to be a methyltransferase that's involved. So, right, 3 plus in some cases the ribose methyltransferase. And that is, in fact, why I left 4 off of this as being one of the possible answers. Because, again, one could argue that you need a ribose methyltransferase and a 7-methylguanosine transferase, which it turns out they are different enzymes that do this. So that's another reason for that. OK, so that's it for capping. Now, splicing. Splicing is a really amazing process, and we're still, we being the scientific community, I don't work on this, but still trying to figure out exactly how it works. And it's a very, very complex process. And again, we're still trying to piece apart. The very basics are what we're going to talk about today, but a lot of other things are still very much up in the air. We don't completely understand them. When splicing was first found, it was actually really kind of a surprise because molecular biology shouldn't be ridiculously inefficient. You shouldn't be chopping out extra pieces of RNA. You shouldn't be spending all of these nucleotides to make the RNA and then chop them out and get rid of them. So it was really kind of surprising. So I wanted to spend a couple of minutes talking about how splicing was originally discovered. And that has to do with what's called the R-loop technique. We talked a little bit about D-loops when we talked about double-stranded break repair. We've got that single-stranded piece of DNA that goes in to an otherwise double-stranded DNA and displaces one of the strands of DNA. It's called the D-loop. Here, you're basically doing exactly the same thing, only it's the RNA which is being used to push apart another strand versus using that DNA that's got Rec A or RAD51 or one of the homologs that's associated with it. So you take your processed messenger RNA. Here, you can tell it's a eukaryotic one because it's got a cap and a tail. You heat up your DNA. The 2 strands come apart. You put it back together, and your RNA will hybridize here. And the hybridization is just like you get with DNA when those 2 strands are going to come back together. And you'll have a little piece at the end that is not hybridized because it's your poly A tail. Poly A tails are not present in the DNA. These are things that get added later. So you have a little tail at the end. And so in this process, you can actually see, here's a nice example of a piece of DNA which has a double-stranded piece all the way along it here and a single-stranded piece relative to it. Now, how the heck do you know it's double-stranded versus single-stranded? This one's a little thicker than that one. And you spend a long time staring at electron micrographs in the process. And then you draw these nice little pictures down here, which makes it a lot easier to interpret. Here is your 3' poly A tail here. And in this case, there's actually a little bit of extra stuff at the 5' that doesn't match with this DNA, which was put here. And this is the first indication that something bizarre was going on here. Because why? Okay, now the 3' end not matching kind of makes sense. It's a poly A tail. But why is the 5' end not matching? And in some cases, you had pieces of DNA. Here's your RNA that, again, it's got a nice little 3' poly A tail. But this 5' end is actually matching a different piece of DNA, which you had in there. So what the heck is going on here? All this DNA should be contiguous. And the classic example of this is here. This is a colorized version of one of the electron micrographs that was in paper, from Rich Roberts' lab. It's a lot easier to see in color than it is in the black and white electron micrograph. And basically what they have here was they took a messenger RNA from a viral protein. And in this case, it was adenovirus, and viruses We talked about this a little bit before. Next week, you can sign up for my virology class and hear a lot more about it. The messenger RNA for the capsid, which is the shell around your nucleic acid, you need a whole bunch of that because you need a whole bunch of the protein to make the shell around your virus. So it turns out that this particular messenger RNA, the hexon messenger RNA, is the most prevalent RNA that's present in a virus-infected cell. So it's actually relatively easy to purify this particular RNA. And then you take the DNA from the virus and do this R-loop technique. So what we have, let's see, get my pointer to actually work here, will be good. We've got here our double-stranded piece. Again, this is our RNA-DNA hybrid with its 3' poly A tail. But up here at the beginning, at this 5' end, there are all these big loops of DNA. This C loop of DNA, this B loop of DNA, and the A loop of DNA. But they're all being held together by the RNA, the 5' end of the RNA. And what that means is that this RNA is complementary to the DNA here and the DNA in these 3 little bits right here, also called the tripartite leader sequence. Again, molecular biology, when we talk about adenovirus, we'll discuss this at length. But this was really good evidence that the messenger RNA doesn't match the DNA. There's something different between your messenger RNA and the template that you started from. At first, people thought this was all boring viruses, who cares, or really cool viruses, but just true in viruses. This was shown first again in 1977, Rich Roberts and Phil Sharp. They both got the Nobel Prize for discovering these things, in theory, independently of each other. That's a whole different story. But very soon thereafter, in fact, both labs showed that this was not just something that happened in viruses, happening in pretty much every single eukaryotic gene. So here are just the examples up here from our textbook. Some genes in our genome have just a couple of exons that are right next to each other, very few introns. Most of the genes in our genome are a lot more like this. Exons, which are these coding sequences, the vertical red or pink lines here, versus the yellow introns, which are getting cut out here. Exons can be as small as 8 base pairs, which is pretty darn amazing. You have this really tiny piece. But usually they're about 145, which is also interesting because that's almost exactly the same size as one nucleosome is being wrapped around. Yeah?
So on the bottom diagram, the colored bits in between the black bands, those would be introns?
So here, this is the DNA which would be coding for the introns. So in this particular case, so this is showing the messenger RNA that gets transcribed from here. But in this case, it's really, what we're looking at here is the DNA, and then the final processed messenger RNA and how it's hybridizing to that. So the loops here are, well, an intron is only true when it's in a RNA sequence, and so it's DNA which is coding for an intron. Yeah?
So like all the loops in like the C loop and A loop, so that's just DNA
That's DNA that has been looped out because it doesn't match to the RNA. So what the RNA is doing is pulling these 3 black pieces here together, together with the green piece at the end, pulling all those together, and what happens to the DNA in the middle? It gets just looped out. And it turns out you can actually measure all of these loops, and that's what they did with the electron micrographs, and we're able to specifically identify exactly where these things were, and that was why Roberts and Sharp ended up getting the Nobel Prize for studying this. So a couple of things, of course, much later than that. People found that the machinery for doing this, taking a pre-messenger RNA or a primary transcript, which is just the RNA before splicing happens. And making it into the final messenger RNA requires 5 RNAs, which is actually really kind of interesting. You need an RNA to process an RNA, and more than 100 proteins. In fact, I think in the textbook it says about 200 proteins, or maybe it's in the literature I've been reading saying 200 proteins. So there's a lot of machinery that's involved in this whole process. It's an isoenergetic process, quote unquote isoenergetic process, because you're breaking phosphodiester bonds and forming phosphodiester bonds, but you're breaking and forming exactly the same number. So overall, it's not something which you need to input energy to. Turns out, however, there's a lot of ATP, which is hydrolyzed, and that has to do with the specificity that we'll take a look at in just a second. Yeah?
So I got 2 questions. In the
Sure.
center of that mess you got there, there's a RNA polymerase 2 pumping out that mRNA, correct? Or is this after the polymerase activity?
Okay, so this is after polymerase activity. This is a completely what we call an in vitro experiment, so done in glass. So you purify the messenger RNA, and it's this hexon messenger RNA, which means there's a whole bunch of it, so it's pretty easy to purify. And then you purify the DNA from the virus separately. And in a test tube, you put them back together.
Gotcha. So when this is actually happening inside the cell, what's happening is you're using DNA to make an RNA template.
Yep.
The RNA template is then matched against the exons of the DNA that is just copied from to splice the RNA pieces together.
Okay, so that is not completely correct. So what happens, sorry, should I repeat that? Everyone's kind of got it? So the question is basically, sorry to repeat this again, so it's for anyone that can't make it today. The idea here is so the RNA polymerase itself makes the whole thing. It's getting actually spliced and copied and all these other kinds of things. Splicing happens as it's being made. But let's just assume that it's made by the RNA polymerase. So the RNA polymerase itself is just going to make this whole long primary transcript or pre-messenger RNA. Then, after that, the introns get cut out. But those introns get cut out having nothing to do with the DNA. And we'll talk about how that happens in just the next couple of slides there, how splicing takes place. And it actually has the only thing it has to do with relative to the DNA is what that DNA is coded in terms of the sequences, which are there. And it's all of these RNAs and other proteins, which are then important for the specificity, where you make the splicing. What's an exon, what's an intron? Because it's all just RNA. How do you know what's an exon and what's an intron? Okay, other questions about this is, this electron micrograph. Basically, it was this figure that got Phil Sharp a Nobel Prize. It's pretty cool that one figure can get you a Nobel Prize. I'm still working on that. So how chemically does this process actually work? Well, it turns out that through a lot of work from a lot of different people, including Tom Cech, who also got a Nobel Prize, who I actually met right around when he got his Nobel Prize, which was kind of fun. I thought about doing a PhD in his lab, but decided not to go there. Is what happens is you have your intron sequence, and in that intron sequence is an adenine residue. And that adenine is completely conserved. You always have to have an adenine there. And since this is RNA, that adenine has a 2' OH. That 2' OH can now loop around to the 3' end of this exon and ends up making a 2' to 5', shown right here, phosphodiester bond. So we already talked about 5' phosphodiester bonds. Now we got 5' 2' phosphodiester bonds that form. Now there's a OH at the 3' end of this first exon, which can now go over to the 5' end of the next exon and cut here, releasing this looped or lariat structure, ending up fusing these two RNAs together. And this is just shown here in a little bit more detail what that lariat looks like. Here again, it's a 5' from the very end of this intron that gets hooked up to the 2' in the middle of the intron. So yellow here is all your intron sequence, so it's also the yellow nucleotides that you have here. And then red is that, also known as the branch point, because it's the branch in this particular structure. So that's the chemistry? Yeah.
So it's a 2' to a 5' on the intron?
Yes, correct. So it's a 2', 5' bond that forms. So could you do this with DNA? No, why not? No OH on the 2'. So you can't splice DNA, at least not using this mechanism. There are DNA splicing mechanisms, but that's more like our double-stranded break repair that we talked about before. So how does the cell know, again, to totally over-anthropomorphize here, where that adenine residue is, because there's going to be a whole bunch of adenine residues in your pre-messenger RNA, and where exons are and where introns are. So it turns out that the consensus, quote unquote, again, Stedman loves these quotes, sequence for introns is not very good. In fact, very few nucleotides are required for getting splicing to take place. The only ones which are absolutely required are the ones in the boxes here. So a GU at the very 5' end of your intron, and an AG at the very 3' end of your intron, and then a adenine again in the middle. The rest of the quote-unquote consensus sequence here has an R for purines, has Y for pyrimidines, a whole bunch of Ys, some Ns. It could be any 4 nucleotides. And basically what this means is if you're just looking at a DNA sequence and say, okay, this is going to get transcribed into RNA, what does that sequence look like? You really don't have a clue if that's going to be spliced out or not, whether it's an exon or an intron. So it's actually really hard to tell from looking at these sequences what's an intron, what's an exon. Yeah?
Does this mean that you have a lot of random splicing activity, or that you have splicing activity that you can't really, that the cell can't really prepare for or predict for?
Yeah, so the basic question here is, do you have a lot of random splicing that, again, the cell can't predict and ends up with even less efficient formation of your messenger RNA than you would already expect? We'll talk about some of the mechanisms why it's actually not as bad as it could be, but that's one of the parts of splicing that we don't completely understand at this point, is to try and do that. Part of the process does actually have to do with the parts of the spliceosome, and particularly the RNAs that are involved with the spliceosome. We'll get to that in just a second. Yeah? So we're just about to get there. So specificity for your splicing, yes, it's not very much that's conserved, but there are these absolutely conserved nucleotides. So how do you recognize these nucleotides? Remember, this is RNA. It's not double-stranded DNA with a nice major group with all of the appropriate nucleotides that are present in it. Being able to bind to specific RNA sequences, what's the best way to bind to a specific RNA sequence? So either you've got a DNA or you've got another RNA that's got a complementary sequence to that. So that's where these snRNAs come from. So small nuclear RNAs, 1, 2, 4, 5, and 6. We'll talk about 3 later. 3 has nothing to do with splicing. But U1, U2, U4, U5, and U6 are absolutely critical for this whole splicing process. And those RNAs are probably most important, particularly U1 and U2, for making these double-stranded pieces and giving you the specificity. You've got one RNA sequence that's going to match with the RNA sequence which is in your intron and exon. And the really important thing here is the boundary. It's the boundary between the intron and the exon, is where you're actually going to make those cuts and how they're going to be put together. So that happens here. It's not just the snRNAs, but they're snRNPs. What is RNP? RNP is ribonucleoprotein complex. So it's RNA and protein. And this is just the example of one of those here, RNAs and protein together. All together, this makes, again, molecular biologists love to call things zomes. So the spliceosome is the RNA and all of the proteins, or the RNAs, I should say, and all of the proteins that come together to make this happen. The important aspects are the HNRNPs, and those are particularly U1, U2, and U6, together with a couple of proteins, and you can tell that they're proteins because one's got a P at the end. It's a branch binding protein, and the other one's a factor, the U2 activating factor. So what happens when you have this RNA? It's being made by the RNA polymerase. You have a cap that gets put on at the 5' end. You're transcribing happily along, 5' splice site, and then your A, where you're going to have that branch, the 2', 5' phosphodiester bond that forms, and then the 3' end of your intron. Branch binding protein and the U2 activating factor are the first two things that associate with your intron. Branch binding protein, again, not surprisingly, binds right at the branch point. U2 activating factor is going to be interacting with the U2 snRNP. So U1 and U2 are conveniently the first two that associate here. U2, not surprising because U2AF, interacts with the branch point. U1 interacts through base pairing interactions with the 5' splice site. So it's the 5' end of your intron, 3' end of the exon. Once these two have associated, then you have U4, U5, and U6. They come as a complex. That's kind of why I put them together right here. Those then associate with U1 and U2 and pull everything together. Once they've been pulled together, now there are multiple rearrangements that happen. And the rearrangements here are mostly having to do with base pairing interactions that get changed. So base pairing interactions between U1 and the 5' end of your intron, U4, U5, and U6 interactions which are changing. And those interactions, again, are mostly base pair interactions. So pulling base pairs apart, that actually requires energy. Helicase. You remember the DNA helicase? What's the thing at the replication fork that's burning the most ATP? It's the helicase. So the RNA helicases in this case are what's really using most of the ATP in this process. So let's take a zoom in at some of these places here. So U1 interacts with this completely conserved GU at the 5' end of your exon. It's got a, and this is of course the opposite strand, so it's an AC in your U1 snRNA. That, in the presence of ATP, you have this RNA helicase, and now these sequences at the beginning of your intron interact with U6, and so it's this helicase interaction that pulls these two apart. The branch binding protein switching out with U2, again, helicase activity, because U2 is bound to itself. Now we have binding here, and then this A gets pushed out. Why is the A being pushed out? Because that's providing that 2' OH that can now go and make the here 2' to 5' phosphodiester, which cuts this structure. This is what it's associated with, again, with U2. U6, you've gotten up there through this first ATPase activity. And then U5 holds the two exons together so that you can have this 3' OH from this exon binding to the 5' phosphate from this exon. This is the cartoon version. This is actually much more what it looks like. This is a paper actually from 2015, now from 2 years ago. where they were looking at the structure of the spliceosome. And we'll look at the actual structure here in the next couple of slides. Here, they don't have the whole structure. They don't have all of the RNAs in here, but they have some of the important bits. Here in purple is your intron. In blue is U2. And probably most importantly here is the U5, small nuclear RNA. In here, you'll notice 3 red dots, or actually it's 4 in the back there. These are metals which are part of the active site here in this particular enzyme. Yeah?
So where is U5? Is that in a complex with...
So U5 is coming here right at the end. So U5 comes into the complex with U4 and U6. But then it gets rearranged through the RNA helicase activity to bring all of this together.
And that's what's holding the exon together at the last point?
At that last point, it's holding them together. And you can actually see that here because U5, it's not labeled on here, but this is your U5 structure here. So this is where your binding site is. It's a classic enzyme which provides a very specific binding site for these very specific, in this case, RNAs. But the important thing here is that this is all RNA. There's no protein to be seen around here. It's all the RNA which has the activity. So it's RNA activity which is driving the spliceosome. So they're RNA enzymes. And we'll talk more about RNA enzymes later on, probably on Wednesday in lecture. Yes?
So the U2AF
Mm-hmm.
also helps with the U2 to help push that 2' OH out and bind to the 5'.
Okay, so the question is basically, so I'll paraphrase here, is what's U2AF doing? U2AF basically seems to mostly serve to get U2 bound to that particular position. So it's not the actual protein that's doing that, it's the protein that's bringing U2 to that position. Okay, so it's really the RNA here which is providing the catalytic site. Now, this is just looking at the RNA, the spliceosome, as I've mentioned before, a whole bunch of proteins as well as these 5 RNAs. This is, as far as I know, let's go to the next slide, the most recent structure of the quote-unquote spliceosome. It actually is missing one of the RNAs, and it's only 37 proteins, which again gives you a bit of an idea of the complexity of this whole structure. Here we've got U5, U6, and the intron, again the same coloration, but all this gray stuff around here is protein. So the business end is right here. Again, it's the U6 interactions. All the proteins here basically seem to be holding those RNAs in the right place. And this is the overview down here at the bottom. Again, this is from Science from a couple of years ago. So we're going to have clicker questions. Everyone get their clickers out. Not quite done talking about this yet. But again, the take-home message here really has to do with the RNAs and those RNAs interacting with, through base pair interactions, with your introns and exons, and providing the catalytic site for this first 5' to 2' phosphodiesterase bond formation, and then the 3' to 5' phosphodiesterase bond formation to finish up. And these are, again, just some of those proteins, and this is about 1/3 of the proteins that are normally associated with the spliceosome. Even getting this kind of structure was still pretty amazing, and again, it's where we're at right now in terms of understanding the whole spliceosome structure. So now, finally, we can have our clicker question, which is, which of the following is thought to contain the splicing enzymatic activity of the ribosome, spliceosome. Oops, not the ribosome. That should be spliceosome. My bad. Spliceosome. U2AF, U1 RNA, U4 RNA, U5 or U6. Ten, 5, 4, 3, 2, 1. Lots of differences of opinion here. So U2AF, we just talked about. It's the activating protein for U2. It's getting U2 onto the intron in the first place. So is that the one that's got the activity? Okay. What's U1 doing? It's not no. It's interacting with the 5' end of the intron. U4, we talk much about U4? Not really, so it can't be U4. Nobody likes U4 anyway. U5 and U6 are what we have left. So what's U5 doing? It's bringing the two exons together. Where were those red dots that I showed 2 slides ago? They were in U6. Can we go back and look at that slide? Okay, yes. So, U6, the green one, has these catalytic enzyme sites right here, the red dots. Catalytic triplex, all in this green RNA. That answer that question?
Yeah, can you go back to the blob drawing?
This one? Yeah.
So the U5 only just holds the exons together. It doesn't have any enzymatic activity.
So the question here is that U5 is just holding the ends together. And that is correct. It does not have the enzymatic activity. The enzymatic activity is all in U6. And that means that this blob diagram is, again, being a 2-dimensional image. And U6 is way the heck over here. It's really this U6, and particularly in this first step here, you can see that this activity is catalyzed, or it's very close to U6. The same thing is true right here. It's not U5 that's involved. U5 is just holding things together. And it turns out the U5 is also very much a negative regulator of U6. It only then is going to be able to do its, U6 is going to be able to do its job when U5 has been separated from it. Yes, sure. BBP. BBP is the branch binding protein. So what the branch binding protein does is it binds to the A in the middle of your intron where you're going to have that taking place and interacts with U2AF, which then brings in U2 and starts this whole reaction. So it's one of the very first steps. Yeah, in the back. Does U1 become U6? No, they're separate RNAs. U1 then, let's take it back up here. U1 is exchanged for U6. And that's where the RNA helicase activity comes from. That's the ATPase that you require to pull those strands apart. Okay, more questions on this? Yeah.
What is
U4? U4 is part of that regulatory complex. And so when you look at where it comes, all these A's, U4, U5, U6 come in all together. it's base pairing with U5 and U6. Once you have that come all together to the spliceosomal complex, then you have the RNA helicase activity that also pulls those apart and allows U6 and U5 to do their jobs. Other spliceosome questions? We already did clicker questions, so we're not going to have another one. Yay. Okay, so getting back to your question about why splicing just doesn't happen all over the gosh darn place. So a couple of reasons for that, again, which we don't completely understand yet. But there are a couple of them, and probably one of the most important is that splicing happens while you're transcribing. So you only have a certain area that can be spliced. So you don't make the whole pre-messenger RNA and then splice it all up. Then you'd have all kinds of trouble trying to figure out where the heck you're going to actually do that splicing at. So as the RNA is coming off, then you need to have the first exon is going to get spliced usually to the second exon, which then gets spliced to the third. It's not going to get the first splicing to the seventh, etc. But then you also have to know where the exons are. And it turns out that there are a number of proteins that bind very specifically to exon sequences. And those are the ridiculously badly named SR proteins. Why are they called SR proteins? Because they've got a lot of serine and arginine in them. So that's where the SR comes from. Lots of serine, lots of arginine. And those then quite frequently are going to interact with these exons. Once they've associated with exons, then they are going to help U2AF. U2AF is going to bind to the branch binding protein. U2 will bind, and then you can put the whole spliceosome together. So it seems to be that U2 association is really, really important. So that's probably why we have a U2AF. The other thing that all of you, I'm sure, remember is that U2 also interacts here pretty close to the 3' end of each of these introns. And what that means is the branch site is almost always quite a long way towards the 3' end of any of these introns. So the way it's always drawn with the lariat structure is sort of that A is kind of right in the middle of your intron, and it's about half of it that loops around, or 2/3, 1/3. That's really not the case. Usually it's very close to the 3' end, and that's because the U2 is associated there through the SR proteins and U2AF. So it's usually a very long loop structure which is forming there. Yeah?
The 3' end of the intron or the exon?
So it's the 3' end of the intron. It's the 5' end of the next exon. The other thing that we see are these even more poorly named proteins, the heterogeneous, which means there's a whole bunch of different ones, nuclear ribonucleoprotein complexes, the HNRNPs, which associate with introns. But just a whole mess of these different proteins. Another way that usually exons are being found is that they're almost always these same size. Again, about 150. Here they have about 200 nucleotides. And we know that if we just look at exon sizes versus intron sizes. And if you add up all of the known exons in humans, C. elegans, and Drosophila, all of those are around a couple of hundred nucleotides in size. Introns, on the other hand, are all over the place. And so this is just plotting these introns. We already talked about the human genome having a lot more introns in it than some of these other model organisms, the C. elegans and the fruit fly. But some of these can be literally tens of thousands of nucleotides in length. And figuring out, again, with this whole system that that's the case, why you want to use one versus another is still a very interesting question. One theory, and this is highly theoretical, which is why I put the question mark after it, is that being bound to one nucleosome, sort of one nucleosome, one exon, does seem to make a little bit of sense. Whether it's cause effect is still not entirely clear. Maybe SR proteins like to bind around on the outside of a nucleosome complex. It's probably not completely coincidental, but nonetheless, it's not entirely clear whether that's the case. Of course, the nucleosomes are associated with the DNA, not the RNA. So there's probably something about transcription, which of course is happening at the same time as splicing, runs into a nucleosome, maybe it slows down a little bit, the splicing has a chance to try and catch up. Again, this is a lot of hand-waving right now in terms of what's going on in terms of all of these. And as if that weren't crazy enough, it turns out that there's not just one spliceosome. There are multiple spliceosomes that are present inside the cell. And they function, fortunately, in terms of the chemistry, everything we're talking about here, basically in exactly the same way, but instead of having U1 and U2, they have U11 and U12. Why are these different? The difference is in the RNA sequence. And if they had a difference in the RNA sequence, now they're going to have different sequences here at the 5' end and also at the branch point and 3' end of your intron. These are relatively rare, but there are some of these. And then in the rarest case, you can actually find RNAs that can be fused together. This is an RNA splicing mechanism. And we actually have two different RNAs that if they've got the right sequences at each end and a very specific protein can allow these two ends to be put together. Yeah?
So the main, could you go back to that?
Yeah, I'm going back to here. Yeah.
The main difference I see in those is the U12 and then what's the little SL down there? And the
Down here or down here?
RNP, the small nuclear
Yeah.
nucleoprotein complex.
Okay, so difference here, I'll step through this again. So the difference between the normal, quote unquote, spliceosome and the U12 type is differences in the sequence and the different HNRNPs. But just the first two. You'll notice down here, U5, U4, U6 are all the same. So it's just those first two that are different. In this trans-splicing case, which is very rare, but just wanted to bring it up, and it's also in our textbook, you have specific binding by this SL-SNRNP, and that's actually a sequence-specific interaction. So in the exon, there's a specific sequence that's going to interact with this SL-SNRNP. There's no U1 there. That brings U4, 5, and 6 together. And then if there's a U2 bound in another piece here, these can get hooked together. But it's only if you have this very specific interaction which is taking place here. Yeah?
So, since they're sequence specific, do they interact at different sites or do they like overlap? Can the U12 type interact at the same sites that the major does, just not as well?
Okay, so the question here is if you have a particular intron, is it going to be treated by just U12 or the major spliceosome complex? And it does seem to be completely different. You either have the major spliceosome complex or the U12 way that it's working. So we'll continue and talk more about alternative splicing on Wednesday.